Today, I want us to have a conversation concerning the RH genetics and RH genes in particular. So, I want you to bear in mind that we have RHD and we have RHCE, which are two closely linked genes that are located on chromosome 1. Yes, chromosome 1. Don't forget it. Chromosome 1. So that is where it is located. Uh, and it controls the expression of the RH protein. So in order for genes to be expressed, it has to be present, whether it be dominant or it be recessive. So what I want you to bear in mind is that you have the DCE classification, and from the DCE we have the different branches of the RH being expressed. And on the chromosome number 1 is where the RH proteins are expressed. So the RHD codes for the presence or the absence of the RHD protein. Just bear that in mind. Keep it simple. We're keeping everything simple. So the RHD codes for the presence or the absence of the RHD protein. So it's like we're saying, is it there? Is it not there? Is it there? Is it not there? So somebody can be positive when it's there, and it can be negative when it's not there. So it's just that simple. So the second thing that I want you to bear in mind is that the second genes, the RHCE, codes for either RH capital C, lowercase e, or what we call RH lowercase c, capital E. So those are two of the combinations that you can expect to get, but it doesn't stop there. You can also have RH lowercase c and lowercase e. And not to mention, we can also have RH capital C or uppercase C and also uppercase E protein. So remember, RH blood group system, we have discussed the DCE arrangement, and we understand that you have the RHD, which code for the presence or the absence of the RHD protein. All right, then we have C and E, and we know that C and E can be in both capital or common form, or uppercase and lowercase form. So a combination of those forms can be expressed when we're talking about the RHCE gene. All right? So that should give you an essence as to this whole dynamics of the RH gene system. As we move a little further, I want you to just bear in mind that the RHD and the RHCE are codominant, which means that they all produce by inherited, typically produce antigen detectable on the RBC. So what are we saying? The RHD and the RHCE genes each have approximately, let's say, 10 exons and are 97% identical. So each gene has a number of alleles, most of which have been identified through molecular testing techniques. So this is where immunohematology and molecular testing comes in handy. And when we intertwine them, then we can have a more thorough understanding as to the different nuances that we have currently in the blood banking system. So when we have genes, when we have expression, when we have multiplication, when we have reproduction, when we have all these dynamics taking place, we can also have something that we consider to be mutation. So a mutation can occur just like with any living organisms. If you mate with people who are too close to you, you can have mutation in your offspring and they may have disease states. All right. If you mutate with someone who has a genes which is dormant, and then in that mutation situation of commingling or breathing, interbreathing, then what can happen is that that genes will become expressed in the offspring that is produced. So just like that, in a normal situation of operation, human interaction, we can have mutation and we can have mutants. Yes, we're not talking about the Teenage Ninja Turtles or the mutants, the team X from Xavier Institute. We're not talking about those kind of mutations. We're talking about regular mutations of regular people, which cause them not to be as is expected by the normal population. All right. So mutations can occur by the addition, the deletion, the duplication, or whatever dissection that may take place of a particular genes, which would cause it to be expressed in a manner that would give us something that we do not necessarily desire to have, or something that is unexpected, right? So numerous mutations have been described in the Rh genes. Greater than 250 alleles have been determined in the RHD genes, and 50 alleles have been found in the RHCE allele, and the number continues to grow as people do more and more research, and what is there becomes more evident to all of us. So a complete list of the RHD and the RHCE genes can be found on the resource database, which is the NCBI, the National Center for Biotechnology Information. So all of these things that we're talking about, as people discover them, as more information becomes more evident and prominent, and more research is done, then this database is usually updated after it has been peer reviewed. And this database also maintains any form of mutations that may be observed. All right. But fortunately for the medical technologists or the medical laboratory scientists or even the clinical laboratory scientists, many of these mutations are rare and do not change the serology observation in day-to-day testing. So even though these mutations exist, they are important to know, they are significant to a degree, but they do not impact greatly or significantly our day-to-day testing of a patient's blood groups. All right. Now, let's get into another section that you may find quite interesting, the Rh-associated glycoprotein, or the RHAG. So another gene important to the Rh antigen expression is the RHAG. This is basically the Rh-associated glycoprotein, and it resides on chromosome number 516. So RHD, RHC, RHE exists on chromosome 1, but now we have the Rh associated glycoprotein, which exists on 5 plus 1 equal 6. So we have chromosome 6 is where it resides. So the product of this gene is Rh associated protein. So when this gene is being expressed and it is produced, we have the RHAG, and that is just a fancy name that has been given to it. So this polypeptide is very similar in structure to the RH protein, with the difference being that it is glycosylated, which means that there are carbohydrates attached. Within the red blood cell membrane, it forms complexes with the RH protein. All right. So the RHAG is termed as a co-expressor and must be present for successful expression of the RH antigen. So this here is a co-expressor, which means that co means just like when someone is in correlation with, they have to be there, the other party have to be there so that they can combine together, right? So if one is there and the other is not there, it cannot really be expressed. So this is basically the co-expression mechanism that is taking place. So just remember that little caveat that exists with the RH associated glycoprotein. So this is something that many persons may find quite interesting and enticing, and it is good for us to have an appreciation as to how the RH blood group system operates. So as we discussed before, it has to be co-expressed. So by itself, this glycoprotein does not, I repeat, it does not express any RH antigen. So basically, it is present but absent. So it is there, but it cannot do anything. So this needs something else to be present in order for it to be activated. But when mutation in the RHAG genes occur, it can result in missing or significant alteration of the RHD, the RHCE protein, which affects the antigen expression. In rare instances, individuals express no RH antigen on their red blood cell because we know that the RH is expressed on the red blood cell. That is why we can use the red blood cell or the cells from an individual, dispense it on a cover slip or on a slide or in a tube, and then add the antibodies to the surface of those cells so the antigens that are present will come together if they are there, and then we have what we call an agglutination, and based on the agglutination it gives us an indication as to what is present, because if there's an agglutination there is an antigen antibody complex formation, and when the antigen antibody complex formation, that there is confirmation of the presence of whatever it is that we were testing for. So it's that simple. All right. So as we get into the meat of the matter, we will get a better understanding and appreciation, especially as it relates to laboratory medicine and the caveats existing in the practice and the application of this system. So this blood group system is critical and important for us to know and understand and appreciate. So as we continue to discuss the RH blood group system, these individuals who don't have any RH antigen on their red blood cell are considered to be RH null. Null mean none, basically. Null mean there is non-existence, right? So they are the null phenotype. And we may have alluded to them previously, so we have an idea as to who they are. But we're going to discuss them further when we get a little deeper into our study. So the RHAG has been associated as a separate blood group system and is discussed in further details and will be discussed with you as it becomes more prudent and relevant for us to build on the information that we would have garnered in this realm. But for now, let us just get the basics down about the RH blood group system, right? Now, let us think about the RH positive phenotype, because we said that someone can be RH positive, someone can be RH negative, someone can be RH null. We understand that, but we're going to talk about the RH positive phenotype and the RH negative phenotype. But let's get to the RH positive so we can move further into the RH negative. So the RH genes are inherited as codominant alleles. So a Rh positive individual inherits one or two RHD genes, which results in the expression of the RHD antigen and are typed as Rh positive. So when we add the red blood cells of the patient on the slide, in the tube, after we have made that suspension, whatever it is, and then we add the antigens, the D antigens, the D antibodies rather, to the cells which has the antigens on the surface, then we'll get a reaction. If it is present, then we will see a positive reaction, which means there is be agglutination, or it would look something like a clumping formation. If it is negative, then it's going to look like nothing took place, nothing reacted. All right, so that there is just what we want to have in our minds. So in addition, the RHD genes, two RHC and E genes are inherited from each parent. So your parents are important in the process of your existence. And we get some things from our parents 50-50, while others we get 100. It would appear, but not necessarily so, because we have to take 50 of the parents, the mother, 50 of the other parents, the father, and then we combine, and then we get the actual organism or the actual entity that you are, right? So all of this is just for us to have an appreciation as to how the RH blood group system is derived. So we're going to get a little bit deeper into some schematics so that we can see how the inheritance occurs. Numerous mutations of the RHD genes have been discovered that have caused weak expression of the RHD antigens. So you may have heard, if you have previous knowledge, you may have heard of weak D, you may have heard of cis strands, and you may have heard of different variations. Those we're going to be discussing, but I just want you to have your mind wrapped about the basic concepts of the RH blood group system. All right, so now let us move into the RH negative phenotype. So when we talk about the RH negative phenotype, if it's positive, it's there. If it's not there, then it's negative. So the RH negative phenotype are also called that because of the lack of its detectability. All right, so if you cannot find it, then it's not there in that moment, right? So that there is exactly what we're saying. So that is why we say it's RH negative. So the words, the terminologies are guides for you to think about so that you have an understanding as to where your mind needs to be when we are discussing these matters. So with that in mind, we can have an appreciation as to the whole dynamics of operation. So the Rh negative individuals can arise from several pathways. So it's not just one single pathway that gives an Rh negative individual. But the most common Rh negative phenotype results from the complete deletion of the RHD genes. That is the individual possesses no RHD genes but have inherited two RHCE genes. All right, so that there is causing some concern, but in actuality it may create a little challenge, but it will not create that significant of a problem if the technologist knows exactly what needs to be done and how to navigate these situations and dynamics. So the blood group of an individual, the race of an individual has something to do with the expression, the prevalence, and the abundance of the RHD genes being expressed. And you must understand by now that in the immunohematology, blood banking, transfusion medicine, a lot of serological testings are done. So even for the Rh antigen, serological testing is done only to define the phenotype. So one may predict the genotype of an individual based on the relevant Rh genes, but determining nation most probable genotype was used in the past. And this is some percentage study. That's a little bit complicated, but it can be still done. Right now, we basically try to simplify things, and we have our DCE system that we use, or the R0, R1 system. That system is the one that we use. You know, we have the Fisher race, we have the Rosenfield system. So those are the systems that we use to determine where we are on this scale. And then we have the international system that is used to determine what the person's blood group is. So there are a few rare Rh antigens, Rh blood groups, Rh blood types that you can find in different ethnicities, and those we can touch on in a little while, so that you have an appreciation that there are rare RZ groups, there are RY group, there are a few, but we're gonna go into them in a little bit more detail in another sitting. But this is just so that you can have an appreciation as to the Rh negative and the Rh positive. The next thing that we would like to make mention of is the D antigen. So the Rh antigen are highly immunogenic, right? The D antigen is the most potent. This is not to be surprising, given that Rh negative individuals lack the entire RHD protein. So exposure to less than 0.1 mils of Rh positive red blood cell can stimulate or initiate antibody production in a Rh negative person. So that there is something phenomenal. And when we have this idea, when we have this design, we can understand why it is so important that the laboratory does the RH typing, why they do the RH confirmation, why they ensure that they're giving the right blood and blood products to the persons prior to transfusion. And we know that RHD has been implicated in hemoblastosis fetalis, or what we call hemolytic disease of the newborn. And this here has been a problem for many individuals. And that is why we administer the ROGAM to individuals who may have the potential of developing antibodies, so that they can be protected to a degree, and their offspring that is not yet but is to come would also have a line of protection. So a lot goes into the testing methodologies and the testing capabilities that the laboratory has to offer. But it doesn't stop there. We have to get a little bit deeper so as we can have the grasping of the diverse nature of the RH antigen system, the RH blood group system. But this is where we're going to pause our sitting in this moment until the next sitting, when we will get down into the meat of the matter of the meat that you came here to devour, as we continue on this excursion of the RH blood group system. Until then, continue to study to show thyself approved. With that said, class is dismissed.